Good day. I would like to welcome everyone to Arcturus Therapeutics First Quarter 2025 Earnings Call. All lines have been placed on mute to prevent any background noise. After the speaker's remarks, there will be a question-and-answer session. If you would like to ask a question during that time, simply press star, then the number 1 on your telephone keypad. If you would like to withdraw your question, please press star, then the number 2 on your telephone keypad. Today's call is being recorded. I would now like to turn the call over to Neda Safarzadeh, Vice President, Head of Investor Relations, Public Relations, and Marketing. Please go ahead.
Thank you, Operator. Good afternoon and welcome to Arcturus Therapeutics Quarterly Financial Update and Pipeline Progress Call. Today's call will be led by Joe Payne, our President and CEO, and Andy Sassine, our CFO. Dr. Pat Chivukula, our CSO and COO, will join them for the Q&A session. Before we begin, I would like to remind everyone that the statements made during this call regarding matters that are not historical facts are forward-looking statements within the safe harbor provisions of the Private Securities Litigation Reform Act of 1995. Forward-looking statements are not guarantees of performance. They involve known and unknown risks, uncertainties, and assumptions that may cause actual results, performance, and achievements to differ materially from those expressed or implied by the statement. Please see the forward-looking statement disclaimer on the company's press release issued earlier today, as well as risk factors section in our most recent Form 10-K and in subsequent filings with the SEC. In addition, any forward-looking statements represent our views only as of the date such statements are made. Arcturus specifically disclaims any obligation to update such statements. And with that, I will now turn the call over to Joe.
Thank you, Neda. It's good to be with you again, everybody. Over the last few months, there has been elevated interest in our mRNA therapeutics pipeline, given that we have meaningful clinical data sets forthcoming. So I will begin today's call with updates pertaining to our mRNA therapeutics pipeline. I will begin with an update on ARCT-032. This is our messenger RNA therapeutic candidate for cystic fibrosis. Arcturus is advancing enrollment in the open-label Phase 2 multiple ascending dose CF study in adults with CF who are not eligible for CFTR modulator therapy or are not taking CFTR modulators due to drug intolerance, poor response, or lack of access to modulators. Each adult participant in the Phase 2 CF study is expected to receive daily inhaled treatments of ARCT-032 over a period of 28 days. The study began last December, and to date, the study continues in multiple subjects without safety or tolerability issues. The company expects to complete Phase 2 enrollment by the end of the year and provide Phase 2 interim data for the first 2 cohorts in mid-2025. I'll now move on to our ARCT-810 program. This is our messenger RNA therapeutic candidate for ornithine transcarbamylase, or OTC deficiency. Arcturus continues to enroll participants in the open-label Phase 2 OTC deficiency study with 5 intravenous infusions of ARCT-810 over a period of 2 months. The company previously completed the dosing phase involving a cohort of 8 people, and the dosing was at 0.3 mg per kilogram of a placebo-controlled European study enrolling OTC-deficient individuals. The company expects to provide Phase 2 interim data this quarter, or Q2 2025. The U.S. Phase 2 study evaluates several biomarkers, including glutamine and ammonia. Elevated glutamine levels can occur when ammonia scavengers are used, particularly in individuals with urea cycle disorders such as OTC deficiency. While ammonia scavengers reduce circulating ammonia, glutamine may still be elevated due to its role as a temporary ammonia repository, and therefore may be used as a useful biomarker to ascertain proper urea cycle function in OTC-deficient individuals. We are also utilizing a newly developed and improved 15N ureagenesis assay. This is supported by a newly published paper out of Haberle's lab at the University of Zurich. This 15N ureagenesis assay is expected to provide important data for monitoring the effect of ARCT-810 in the company's clinical development program. We are encouraged by the comprehensive data we have collected to date with our CF and OTC programs. And given the current market conditions, we have made a strategic decision to focus our resources toward our mRNA therapeutics pipeline. I now shift our attention to our partnered COVID-19 vaccine program. We are pleased about the recent EU approval of KOSTAIVE. This is our self-amplifying mRNA COVID-19 vaccine. Arcturus received an initial milestone payment from CSL, our global vaccine partner, in relation to the EU approval of KOSTAIVE. We continue to make progress expanding the global KOSTAIVE franchise. The company anticipates a marketing authorization application, or MAA, filing in the United Kingdom in Q2 2025, followed by a US BLA filing in Q3 2025. The WHO is expected to announce the updated COVID strain later this week, and we, along with our partner CSL Seqirus, will update KOSTAIVE accordingly to support Meiji's distribution efforts in Japan this upcoming season. Our STARR self-amplifying mRNA platform continues to benefit from meaningful publications. The company recently published a comprehensive analysis of safety data for KOSTAIVE with a 12-month follow-up from the pivotal clinical study in Vietnam, which had over 17,000 participants who received at least one dose of the study vaccine. The study confirmed the favorable reactogenicity profile. Acceptable tolerability of ARCT-154 was also observed in older participants and individuals at high risk of severe COVID-19 due to underlying medical conditions. Long-term data from this large trial suggests that the sa-mRNA COVID vaccine is safe and well tolerated and did not include any reports of myocarditis or pericarditis. In April, Arcturus' Japanese partner, Meiji Seika Pharma, published an analysis characterizing the distribution and clearance of ARCT-154-encoded spike protein and non-structural proteins in the lymph nodes and injection site muscle in mice following a single intramuscular vaccination. The study showed the encoded spike protein reached its highest level approximately 3 days after vaccination and quickly disappeared from the injection site muscle. The spike protein persisted up to 28 days in lymph nodes after vaccination, and the data suggests that this prolonged spike protein expression may be credited for the observed higher immunogenicity. And as expected, the study also confirmed that the replication is limited. Now moving to ARCT-2304. This is our sa-mRNA vaccine candidate for Pandemic Influenza A, which is also known as H5N1, or the bird flu virus. In April, Arcturus received U.S. FDA Fast Track designation for ARCT-2304. As a reminder, this project has been supported in whole with federal funds from the HHS, ASPR, and BARDA. The company recently completed the recruitment of 212 adults, which included 80 participants over the age of 60 years old, in a randomized placebo-controlled phase 1 trial being conducted here in the U.S. The company expects interim phase 1 data in the second half of 2025. And with that, I will now pass the call to Andy.
Thank you, Joe, and good afternoon, everyone. The press release issued earlier today includes financial statements for the first quarter of 2025 and provides a summary and analysis of year-over-year and sequential financial performance. Please also reference our most recent Form 10-Q for more detail on the financial performance. I am pleased to announce we were able to extend our cash runway till the first quarter of 2028. However, it was not an easy decision. Due to the current market environment and uncertainty regarding our regulatory process, we have made the decision to focus our research and development expenditures on our CF and OTC programs exclusively. I am happy to announce that we have received the initial milestone payment from CSL in relation to the EU KOSTAIVE approval. We anticipate an additional milestone payment and will provide an update on our second quarter call. I will now provide a summary of our financial results for the first quarter of 2025. For the 3 months ended March 31st, 2025, revenues were $29.4 million compared to $38 million in the same period of 2024. The decrease primarily reflects lower development milestone revenues recognized under the CSL collaboration agreement as KOSTAIVE transitions from the development stage to commercialization. Research and development expenses were $34.9 million for the 3 months ended March 31st, 2025, compared to $53.6 million in the comparable period last year. The decrease in research and development expenses was primarily driven by lower manufacturing costs associated with the KOSTAIVE, LUNAR-FLU, and BARDA programs, partially offset by increased manufacturing and clinical costs for the LUNAR-CF and LUNAR-OTC programs. R&D expenses also declined sequentially from fourth quarter 24 by almost $9 million. We anticipate additional quarterly declines in the second half of fiscal year 25. General and administrative expenses were $11.3 million for the 3 months ended 2025, compared to $14.9 million in the comparable period last year. The decrease in general and administrative expenses was primarily attributable to reduced share-based compensation costs. We expect general and administrative expenses to decrease during the next 12 months, driven by lower share-based compensation costs and a reduction in expenses related to the commercial transition of the COVID program to CSL. For the 3 months ended March 31st, 2025, Arcturus reported a net loss of approximately $14.1 million, or 52 cents per diluted share, compared with a net loss of $3 million, or $1 per diluted share, in the 3 months ended March 31st, 2024. Cash and cash equivalents and restricted cash were $273.8 million as of March 31st, 2025, compared to $293.9 million in December 31st, 2024. This $20 million decline in cash is reflective of our annualized cash burn this fiscal year. As stated earlier, I am pleased to report that the cash runway now extends into 2028 with the reallocation of resources to the CF and OTC programs, which include significant cost reductions. In summary, the company remains in a strong financial position and has the cash runway needed to achieve multiple near-term value-creating milestones for the therapeutic programs. We look forward to another exciting year with multiple upcoming clinical data from our CF and OTC programs. I will now pass the call back to Joe.
Hey, thanks, Andy. Arcturus continues to make excellent progress across our therapeutics pipeline, and we look forward to multiple key clinical data sets across our mRNA therapeutics and our partnered vaccine programs this year. So with that, let's turn the time over to the operator for some questions.
Thank you. As a reminder, if you would like to ask a question, please press star 1 on your telephone keypad. You may remove yourself from the queue at any time by pressing star 2. And once again, that is star 1 to take a question. We'll take our first question from Lily Sanago with Leerink Partners. Please go ahead.
Hi, good afternoon. This is Lily Ansago from Leerink. Thank you for taking the question, and thank you for the update this quarter. I just had two questions. So the first one, could you provide maybe a little more color in terms of the changes that have taken place in order to be able to extend the cash runway, as well as, the potential incoming cash flows that you anticipate within this new guidance? And then the second question, relating to the development of the KOSTAIVE vaccine, how should we think about potential milestone related to UK approval and US, as well as the timing for those? Thank you.
Go ahead. Sure, Lily. Thank you for that question. It was not an easy decision, Lily, but as you know, in this environment, I think it was very prudent for us to focus on our two most critical programs. And as a consequence of that, we had to make some tough decisions with respect to cost reductions, including, some elimination of early development and R&D programs, as well as, consolidation of, our facilities. So a combination of all these factors, contributed to the extension of the runway, but more importantly as well, you can probably, surmise that guidance we originally provided remained relatively conservative based on, our cash burn historically. So hopefully you'll appreciate that we tend to be hopefully under promising and over-delivering with respect to our cash runway. Hope that answers your question.
It does. Thank you. Go ahead.
You also asked a second question with respect to milestones, potential milestones associated with the UK filing or US filing, and there's no milestones associated with those. Andy, you want to comment further?
We have a milestone associated with the first US, revenues from KOSTAIVE, but we're not anticipating that, Lily, till 2028, frankly, assuming approval in, 26 or so with the US BLA filing this year. So we're not including that in our forecast. Hopefully you'll appreciate, that we'd like to remain conservative with respect to that milestone.
Thank you.
And we'll next go to Yasmeen Rimi with Piper Sandler. Please go ahead.
Good afternoon, team. Thank you so much for all the updates. Wanted to spend some time, as many investors are very much looking forward to ARCT 032's interim readout here mid-2025. I just want to get a good understanding as we get into this mid-readout, now that we have a little bit more granular color, and the press release that's noted 2 doses are going to be provided. I guess, how large could the initial interim cohort be across these 2 doses? What's sort of the bar that for success you want to see? And then to the extent you can talk about the patient population that you're recruiting for this study, that would be really helpful. Sorry, these are like sort of 3-pronged questions along the interim readout. Just want to have a good understanding of what we're going to be getting at that interim look.
No, good questions, Yaz. We're also excited about this mid-year update for our CF program. There's considerable interest in it. It's a great commercial opportunity, and it's an exciting program. But you asked about how large of a data set to expect. The entire phase 2 study has been listed at clinicaltrials.gov as 12. So because this is an interim data cut of drawing from 2 cohorts, you would expect in that 6 to 9 region of subjects. So with respect to the bar of success, clearly this is 28 daily administrations, so it's a significant hurdle to have reasonable safety and tolerability achieved. In addition to that, we'd be looking for FEV or lung function improvements, and the bar that has been established recently is 3%. We believe is if we achieve that would justify advancing the program. Clearly, the higher percentage that we would get would impact the size and nature and cost of our phase 3 trial. In terms of the types of patients, I think you might be inferring to maybe Class 1 and non-Class 1. It would approximately half of these subjects would be Class 1 subjects and the other half would be non-Class 1. But every single one would be similar in that they're all non-modulator responders. And so we don't tend to bifurcate the data. We view this group as having similar unmet medical need and presentation. So and that's it.
That's very helpful. Thank you, Joe. I'll jump back in the queue.
Thanks, Yes.
We'll next go to Seamus Fernandez with Guggenheim Securities. Please go ahead.
Hey, guys. This is Evan weighing on for Seamus. Thanks for the questions. Just following up on cystic fibrosis, we've seen some tolerability issues from a competitor program. Can you remind us again the difference of 032 versus some of the other competitor mRNA programs? And just to hone in on that a little bit more, in terms of the dose range you're exploring and what we may see from these 3 dose cohorts, especially relative to what you studied in healthy volunteers and the initial CF patient studies. And then, in terms of the first 2 dose cohorts, how should we be thinking about that? I guess whether you expect each dose cohort to be efficacious or whether you expect a dose response here. Thank you.
Yeah, a lot there. All good questions. First of all, our therapeutic is different from our competitors. I think the key points of differentiation would be, number one, our delivery technology. Our LUNAR lipid nanoparticle delivery technology is exclusive to us. We discovered this and are applying it to the CF program, and it's chemically different from what everyone else is utilizing. And we've showcased these differences in preclinical animal models. So clearly, there's something there with respect to our delivery technology. When you're talking about safety and tolerability, you also have to recognize the level of purity of an mRNA construct, and Arcturus has significant intellectual property pertaining to the purification of our mRNA, and how this could potentially impact safety and tolerability is on removing impurities effectively. If we do that, then that would logically enhance or expand the therapeutic index and improve a safety and tolerability profile. And those would be 2 key areas of differentiation. We mentioned that there's 2 cohorts, and each of those is attributed to a different dose level. We have, of course, flexibility built into this phase 2 study to have a 3rd cohort or 3rd dose level or expand the 2nd level. We haven't provided any guidance on the nature of that 3rd dose level, but the data that we intend to share mid-year will involve we'll be drawing from 2 different dose levels. And none of these levels of dosing is wasted, we believe. You asked if all of these could be efficacious, and somewhat that may indeed be the case, but we'll save the details and then for that disclosure for mid-year.
Got it. Thank you.
Thank you. And next we'll go to Miles Minter with William Blair. Please go ahead.
Hi, this is Jake on for Miles. Thanks so much for taking our question. Couple for you. First on CF, we're just wondering if you guys have talked about any sort of accelerated approval pathway for ARCT 032, given the unmet need there in null mutant patients. And then a second question on OTC, you guys mentioned measuring glutamine as a biomarker there. Just wondering if you have any sort of initial bar for success where we should expect a percentage of normal on glutamine. Thank you.
Oh, great questions. First of all, with respect to some sort of regulatory acceleration, whenever you see excellent phase 2 data, that would always warrant a discussion with regulators to accelerate the approval pathway, especially like you mentioned for serious diseases where there's significant unmet medical need. We have not shared any data, so we don't know. It would be too early for me to guide whether this is good or great or excellent data. But to simply address your question, if it's considered excellent data, then yes, we would be looking to accelerate the clinical path for sure. With respect to glutamine, I think I'd like to remind everyone listening that if you have a dysfunctional urea cycle, then your body cannot convert that ammonia into urea. So what does it do? It converts ammonia into glutamine. So glutamine is just a great biomarker that can help us understand this disease. In terms of what would we be looking for in glutamine levels, it would be simply to restore normal levels. So very often it's well known that folks suffering from urea cycle disorders, including OTC deficiency, have elevated glutamine levels. And if we could restore those into the normal range, that could be some convincing evidence that our therapeutic.
And next we'll go to Whitney Ijem with Canaccord. Please go ahead.
Hey guys, thanks for taking the questions. Just sticking with CF, can you talk about the time points that you're measuring FEV in the study and what the duration of follow-up, I guess, we should expect from both cohorts? I'm, curious if there's interim time points you're looking at between the 0 and 28 days, and then if there's subsequent time points, and if we should expect all of those for both cohorts in the upcoming data.
Yeah, the details pertaining to this trial design are available on clinicaltrials.gov, and also the CF Foundation tracker that tracks not only our phase 2 trial and the details there, but also our competitors, and that's a good reference for people to look at. We are going to be measuring FEV, or lung function improvement, before this 28-day treatment cycle. So what do I mean by before? Well, there's a screening process where FEV is measured. Then, of course, at day 0, prior to first administration. And then there's a few measurements throughout the cycle, and then, of course, afterwards. It's approximately 7 FEV measurements taken. We haven't shared the specific details of that, just for competitive purposes, but there's plenty of data points in the study to give a decent level of conviction to advance this program further into development. Did I address your question, Whitney?
I think so. For the folks, just to clarify though, so we should expect the full 28 days and then maybe some additional follow-up from both cohorts, or is it that? Well, okay.
Maybe not
Perfect.
The entire follow-up, because these can be a few months in nature, the follow-up. Yeah. So we may not have the entire follow-up for everyone.
Right.
We intend to have safety, tolerability, and the lung function values for the complete treatment course, correct?
Okay, perfect. Excellent. Okay, and then just to follow up, to clarify on the U.S. KOSTAIVE milestone, is it that there is not a milestone on approval, or that you are not including a milestone on approval in your guidance to be conservative?
Okay, yeah, there is a milestone associated with first commercial sales in the U.S., and that milestone is not included in our guidance because we're not anticipating first commercial sales until 2028. And so hopefully that gives you some perspective as to, the cash runway does not include a U.S. milestone related to first commercial sale.
Okay, and also not one related to U.S. approval.
Right, there is no milestone with U.S. approval. It's related to sales, correct.
Okay, perfect. Thank you very much.
Thank you, Winnie.
Thank you. And next we'll go to Pete Stavropoulos with Cantor Fitzgerald. Please go ahead.
Hi, this is Samantha on the line for Pete. Thanks for taking our questions. So on the CF program, given that the impaired CFTR function impedes mucociliary clearance and mucose plugging, do you see potential for O32 to normalize or remodel the lungs? And if it can you speculate on the length of time this may take? I'm curious to hear your thoughts.
No, it's a great question. We're hoping to be the first to answer that question with data. But just some basic comments is that these patients that are participating in our trial do not have 100% lung function, and some of that dysfunction is attributed to irreversible damage. So there's going to be some portion of the lung that just isn't recoverable. But we're also optimistic that there is a portion that is. And so the lung is very well known to regenerate and heal effectively if you can reverse the dysfunctional biology. So we hope to see that in our upcoming trial results. In terms of the time, because this is a topically administered agent, the time course to restoration, it may be different from systemic treatments like you see in the modulator business. We're different. We're topically administered. So accessibility to these cells is going to be a key marker of success. And as the lung heals, then more cells would be accessible. So at least theoretically, we anticipate a steady improvement over an extended period of time. But what is just unknown at this point is how long is that time period and how much material is required. But we're anxiously awaiting the data.
That's very helpful. And one short follow-up question. Besides FEV, are there any other measurements that you could or will look at that might enhance the investigator's conviction in the program, something like quality of life measurements or others?
Yeah, there's a survey that we're provided. There's a validated CF questionnaire that over the last couple decades in the modulator field. They have improved these questionnaires significantly, and that will complement our FEV data. In terms of other mechanisms like mucociliary clearance or others, the FDA came back with some simple guidance there that all they need to see is FEV to justify advancing this into a phase 3 trial. But we are supporting that data set with some questionnaires to see if we can support what we observe in with respect to lung function improvements.
That's great. Thank you guys so much.
Thank you, Samantha.
Next, we'll go to Tom Schrader with BTIG. Please go ahead.
Good afternoon. Thanks for taking the question. You've commented that the next CF trial is going to depend a lot on the size of the efficacy signal. But do you have a sense of the minimum safety database for an indication of this? Is 100 about right, or are you flying sort of completely blind at this point?
We're definitely not completely blind. I think 100 is reasonable. Unfortunately, it depends on how successful the phase 2 results are. If the more successful they are, the more leverage we have to decrease this number and get this into real subjects commercially as fast as possible. But 100 is reasonable. So that would imply that we had 39 subjects in phase 1 and phase 1B. We had another dozen. That would get us over 50 after phase 2. That we'd probably need a minimum of 50 people to strengthen the database to achieve that 100. But we have not, just to be clear, we haven't received that. But that's a reasonable estimate.
And then in OTC, it seems like there's two deliverables. One, you're helping OTC, but in a grander plan that you're getting stuff into the liver. Is that the same readout? Or what I'm really asking is, do you have any way to monitor uptake into the liver that isn't correction of OTC, or is it really the same readout for both questions?
No, there's not going to be a biosampling or some sort of tissue removal as part of this process. The FDA doesn't require that for these significant rare diseases. But we do have a biomarker strategy. We've alluded to glutamine, for example, as one of those biomarkers. But then there's also a functional readout, and the 15N ureagenesis assay is also of interest there. A great paper just came out last week out of the University of Zurich that I encourage people listening to refer to. But that's another a new exciting way to track disease progression or healing.
Okay, great. Thank you.
Thank you, Tom.
Next, we'll go to Yigal Nochomovitz with Citi. Please go ahead.
Yeah, hi, thanks. I had one question on CF and one on OTC. on CF, I think in the past you've talked about some thresholds for FEV1 improvement that you would feel would be sufficiently strong to move forward. I'm wondering if you could comment on that with respect to the upcoming data. And then on OTC, you mentioned some biomarkers. You mentioned the N15 assay. I'm wondering, are you also going to measure orotic acid and uridine? Because those are also urinary biomarkers that would be interesting. And you mentioned glutamine, but I think alanine is another one that could be interesting as a biomarker in lieu of ammonia, which you can't measure. Thanks.
Good. Thanks, Yigal. I'll first start with the thresholds in CF. These numbers that have been communicated over this last quarter especially are based on historical precedents, but in the modulator space. But if you can have a 3%, if you can show a 3% improvement in FEV over a treatment course, then in order to reprove that in a phase 3 study would require potentially hundreds of people. And so that's a significant, endeavor. Nonetheless, one that we would pursue in order to prove that this is real. But if the FEV value goes up to 5%, then your phase 3 trial could be as low as 50 people. Definitely, or at least confidently below under 100 people. So and if you go north of 5%, then in higher FEV responses, it wouldn't change the size of the phase 3 trial, because earlier on this call there was a question about how many people would you need to establish a significant enough safety database, and 100 is approximated at this point. But so whether you see higher levels of FEV, you'd still need at least 50%, we estimate, or 50 participants in the phase 3 trial. Shifting to the OTC program, orotic acid is a biomarker we are tracking as well, and that's in the urine, and that is associated with the urea cycle and hyperammonemia. And then also there is several amino acids and biomarkers that we're exploring. Glutamine is just more well understood and frequently used by clinicians and doctors to track people on ammonia scavengers to look at glutamine to see if that's elevated as well. Then they still have some challenges with hyperammonemia that's outside of their ammonia scavenger medicine and their diet. So glutamine is a more common biomarker that's more well understood in the community. But you're absolutely right. There's other biomarkers we're measuring as well.
Okay, thanks. And then just going back to the 3% versus 5%, what are you assuming there in terms of the dispersion of the data, given it's obviously a relatively small sample size, 6 or 12 people? So what does that mean in terms of how much dispersion you're willing to accept and be comfortable with, whether it's the 3% or the 5%, because obviously that will impact the way you power things.
Yeah. Of course, we'd like to see elevated consistency in small data sets such as this. We'll have the opportunity to share that data and discuss it, but at this time, it's just too early to comment further on that. But yes, to address the core of your question, a more consistent data set would be preferred.
Got it. All right. Thanks so much.
Next, we'll go to Yanan Zhu with Wells Fargo. Please go ahead.
Oh, great. Thanks for taking our questions. So on cystic fibrosis, I thought previously the guidance was data second quarter. Given that the guidance is mid-year, is it okay to assume that more likely the data will be in the third quarter? And what might be the venue for the update? And on efficacy, I was wondering, in terms of, you talk about the bar for FEV1. I was wondering, what is the bar for proportion of patients meeting that 3% minimum threshold of improvement? Would it be if the response rate, if we can call them that, is expected to be fairly broad, or could this be even like a 20% response rate still considered a worthwhile product to move forward with? Thanks.
Yeah, thanks, Yanan. You bring up a great question. With respect to the bar of a response rate, that hasn't really been established by the field. We're going to have to make a call based upon this data, the FEV, and the consistency of it. There isn't some sort of feedback we've received from the FDA, for example, on this question. We have received feedback from the FDA that 12 subjects may very likely be sufficient to advance this. So as long as there's some level of consistency within these 12 subjects, and remember multiple dose levels will be evaluated within this group, then that is likely considered success, and we'll be able to receive approval to advance this into a phase 3 trial. Then you asked another question, though. It was about
the timing and venue.
Oh, the timing and venue. Yeah, well, because this data set is involving data from 2 cohorts now, we wanted to allow that extra few weeks just to schedule and get additional numbers to strengthen the data set. So it's just that's the reason for focusing on a mid-year interim data update. In terms of a venue, we haven't decided on that, but that is something that we'll communicate relatively soon here.
Okay. If I may, also wondering, since you are moving ahead with filing the BLA, so you're still on track for filing the BLA for KOSTAIVE in the U.S., I was wondering, with the new leadership at CBER, have you had any interactions with FDA very recently, and what is the expectation for this filing? Thanks.
Our interactions with the FDA, I'll first comment on that, have been normal. The feedback we've received has been within the expected timeline or even earlier in the example of the fast-track designation on the vaccine side. So we haven't seen any changes to regulatory environments for at least our mRNA-based vaccines and therapeutics. We do recognize that there's a passionate new administration, but all of them and all of the employees we've talked to at each of these agencies are passionate about gaining access to safer and better medicines and vaccines. And that is almost a credo at Arcturus. We want to provide that, and we're providing data to support that notion. So I think the science will stand on its own legs throughout this process.
Great. Thanks for taking the questions.
Yeah, thank you, Yunan.
We'll go next to Yale Jen with Laidlaw & Company. Please go ahead.
Good afternoon. Thanks for taking the questions. Just two from us. The first one is for the ureagenesis asset you mentioned during the call. Could you elaborate a little bit more in terms of any specific readout that can, very relevant to the data release? And maybe just the second question really here is that on the big picture perspective, that given that you are focusing much more on your in-house programs, and so far you have 2 very active ones, would you consider a little bit more, maybe even in the early stage, so maybe enrich the pipeline, maybe sometime come out later this year or late next year to enrich that? And thanks.
Yeah, the early stuff does not consume a lot of resources. So, clearly we want to be in a position of being able to announce new programs when it's appropriate upon establishing any sort of proof of concept for each of these therapeutic programs. And we've already done a considerable effort there already to date. So we're in a position to move if needed. Our focus on our budget and extending the runway hasn't impacted our subsequent programs. That work's already been done.
Okay. In terms of the ureagenesis asset, any specific readout we should pay attention to?
Yeah, I encourage people to go to the 15N ureagenesis paper that I've highlighted a couple times on this call out of the University of Zurich. It's new. It comes out of the Journal of Metabolic Health and Disease, and the title of it is Characterization and Treatment Monitoring of Ureagenesis Disorders Using Stable Isotopes. But we just believe it's a significant upgrade on the ureagenesis assays of decades ago. It was a considerably better process, and we'll be collecting data within the context of what's been utilized in this paper.
Okay, great. That's very helpful.
And we'll provide more details on this at the right time. We've just planted that seed. If that makes sense.
Okay. Well, great. Thanks a lot, and congrats on all the progress.
And this concludes our question and answer session. I would now like to turn the call back over to Joe Payne for final and closing remarks.
Hey, thanks everyone for participating on the call. I know there's some conferences up and we can meet face-to-face, but if there's any remaining questions, don't hesitate to reach out to our team and we'll get back to you. Thanks, everybody.
Thank you, and this does conclude today's conference. We thank you for your participation. You may disconnect at any time.